So now we're going to talk specifically about genetic disorders, even though we talked a little bit about it in the last section. And of course, when I divide, the idea of my dividing stuff up is that I'm going to make each one shorter, and I end up blabbing on and making them longer anyway. But you know that about me, so let's just move on. So single gene disorders, there are more than 6,000 abnormalities that are known with a single gene disorder occurring in 1 in 200 live births. So that's a lot. That is, very high prevalence. Usually single gene disorders are classified by inheritance patterns. They're going to be either recessive or dominant, or X-linked recessive. So when we say recessive or dominant without the X-linked, they're autosomal recessive or autosomal dominant, or an X-linked recessive. So in single gene disorders, a single gene controls a specific function. So the non-pathological example is color blindness. The pathological examples that have more widespread systemic effects are cystic fibrosis, Tay-Sachs disease, and phenylketonuria. Cystic fibrosis, the gene affecting it is on chromosome 7. Tay-Sachs disease is on chromosome 15, and phenylketonuria is on chromosome 12, I believe. So even though these particular disorders have been known for a pretty long time, it's been only recently that the genome has been mapped to determine exactly what chromosome is affected. So that could have potential for treatment in the future, which is pretty cool, I think. So cystic fibrosis, it's an inherited genetic disorder that's the gene's located on chromosome 7. It causes really thick, tenacious mucus to be excreted from the exocrine glands. And there are systemic effects from this. The primary effects are seen in the lungs and the pancreas, even though there are effects in other areas. In the lungs, mucus obstructs airflow in the bronchioles and the smaller bronchi. So that's a huge issue right there. It can cause permanent damage to the bronchial walls, and infections are common. So the type of infections that are common in people with CF are caused by Pseudomonas aeruginosa, which is everywhere. It's in the soil, it's all over the place. And Staphylococcus aureus, which is one of our normal flora in our body. So it's one of those opportunistic pathogens where it gets in the wrong area and it causes an infection. People with CF are really prone to infections, so it makes it very difficult for them. They also have effects in the digestive tract. Meconium ileus. Meconium is the sort of the first fecal matter in infants. It's the infant liver is starting to produce bile, and it's starting to be combined and excreted in fecal matter. So meconium ileus is where the meconium is super, super thick in infants with CF, and it can cause a small bowel obstruction in the ileum. It can also cause, the cystic fibrosis can also cause blockage of the pancreatic ducts, so that is going to affect digestion and breakdown of nutrients, obstruction of the bile ducts, which also causes problems with digestion and breakdown of nutrients. And the salivary glands are often mildly affected. In the reproductive tract, you can get obstruction of the vas deferens in males and obstruction of the cervix in females. In the sweat glands of someone with CF, there's a high sodium chloride content. So before there was genetic testing available and people knew that they had the gene for CF in their genetic makeup, a lot of times they would be, the first thing that the mother would notice was that the baby tasted salty. When you kissed them on their forehead or something like that, they tasted salty, and so they would do a sweat concentration test to test for that. Now there's genetic testing, and a lot of people who know that cystic fibrosis runs in their family get tested, and they know they carry the gene with the potential for having someone with cystic fibrosis. So this is from the book And this is from chapter 13. They actually talk about cystic fibrosis in the respiratory chapter, but I put it to the genetic chapter because it is a genetic disorder. So it's an autosomal recessive disorder, meaning that you can be a carrier. You have one good copy of the gene, one bad copy of the gene, and your child can get either. So both parents have to be carriers in order for you to potentially have someone who has two bad copies of the gene and develops the disease. So this flowchart shows the effects on all the different parts of the body. So the signs and symptoms that show up, the meconium ileus, which is that small bowel obstruction from the thickened meconium, salty skin, and that might lead to performing a sweat test and the diagnosis of cystic fibrosis if you don't already know. Signs of malabsorption. Steatorrhea is like a fatty, bulky bowel movement, and abdominal distension. So they're not absorbing the nutrients in their food because of the blockage of the pancreatic and bile ducts. Chronic cough and frequent respiratory infections that tend to increase over time, and failure to meet normal growth milestones. So cystic fibrosis used to have a very poor prognosis for living beyond the age of, probably mid-20s. But now that there are more treatments available, people are living longer with cystic fibrosis, and I think that's great. So it's usually diagnosed by the sweat test, genetic testing, testing of stool, radiography and pulmonary function tests, and blood gas analysis. Because of the improved treatments with an interdisciplinary approach, people are living longer, and this is great. Replacement therapy for pancreatic enzymes and other enzymes and a well-balanced diet can really help to minimize the symptoms. Chest physical therapy or physiotherapy if you live in Canada. That's one of the things that you'll learn in your special topics class is postural drainage. You use it for other things besides cystic fibrosis, like pneumonia and some other things. But one of the things that we might be doing is teaching the parents how to do chest therapy on the child and how to do that postural drainage to help clear the mucus secretions in the lungs. A lot of people who have kids with CF will homeschool their kids so they don't get exposed to more pathogens in school, which is probably a good idea. There are some interesting devices, like there's a little vest that you can put on that vibrates the chest and breaks up those secretions and helps clear them. In PT, we might be working on things like jumping on a trampoline, doing activities that are sort of jostling type activities to break up that mucus as well. So lots of good hope for cystic fibrosis. One of the world specialists on CF is at Seattle Children's Hospital, and so there, because the treatment is getting better, people are living longer, which is great. There is a link to a news story that's from Whatcom County of someone with cystic fibrosis who ended up having some transplant surgery and was running a half marathon in the article. So interesting stuff. So there's hope for those things. Other single gene disorders, so that CF is an autosomal recessive. There are autosomal dominant disorders where if you have one copy of the gene, it will be expressed. That includes adult polycystic kidney disease, Huntington's chorea, familial hypercholesterolemia, and Marfan syndrome. So this picture is the comparison of someone with Marfan syndrome, their hands with someone who doesn't have it, and people tend to have increased length in the long bones. It's a connective tissue disorder, mostly, Marfan's is. And some of the side effects can include aortic dissection because the connective tissue holding the arteries is weaker. and that can kill you. So some of these autosomal dominant disorders, all four listed here, are considered, a lot of times they're not, you don't see the effects until you're an adult. And so they're considered a delayed lethal genotype, meaning you don't know you have it until you've likely already passed on the genes to the next generation. So that delayed lethal genotype kind of hides until the symptoms become obvious later in life. And we'll talk more about Huntington's in neuro next winter because it affects the basal ganglia. So autosomal recessive disorders, both parents have to pass on the allele for the disorder. So we talk about either heterozygous or homozygous. Heterozygous means you have one good copy of the gene and one bad copy of the gene. Homozygous means you have either two good copies of the gene or two bad copies of the gene. So with genetic disorders, we'll talk about heterozygous, unaffected, and homozygous, affected or unaffected. So when it's recessive, you have to be homozygous with two bad copies of the gene to be affected. Because it's an autosomal disorder, male and female children are affected equally. So someone who is homozygous recessive or homozygous for the bad gene has the disorder. And someone who is heterozygous has no clinical signs of the disease, but they are a carrier, so they could pass it on. And then someone who's homozygous for the dominant form of the gene is not affected and also can't pass the gene on because they have two good copies of the gene. So in a lot of these charts, it's sort of easy to think that, oh, the couple has 4 kids and they have a 25% chance of being affected, 25% chance of being unaffected, or, one kid's affected, one's unaffected, and 2 are carriers. But really, each time they have a child, that child has a 25% chance of being homozygous recessive, and therefore being affected, homozygous dominant, therefore being unaffected, or heterozygous unaffected and they are carriers. They have a 50% chance of that. So each time they have a child, those are the odds. 25% affected, 25% unaffected, 50% chance of being a carrier. So it's easy to think that, like, oh, we'll just have 4 kids and then only one of them will have the disorder. But that's not how it works. You could actually have, like, say you have 2 kids, they could both have the disorder because each one has a 25% chance of inheriting the 2 bad genes from their parents. So each kid is, like I said, it's another iteration of the same genetic experiment. And the same chances exist for each gene. So those are the ones that are difficult. If you're heterozygous and unaffected, you're a carrier. And so then if you marry someone who's also heterozygous and unaffected, you're both carriers, and each child you had has a 25% chance of being affected by the disorder. So cystic fibrosis is an autosomal recessive, carried on chromosome 7. Fetal ketonuria is carried on chromosome 12. It's an autosomal recessive. It's a defect in protein metabolism. And the metabolites build up and can cause developmental disorders. They cause brain problems. So they test for fetal ketonuria at birth, and if you know you have it at birth, you can make dietary changes that prevent problems. So the more that's known about these, the more problems can be prevented. Tay-Sachs disease is an autosomal recessive, and it actually affects, it's on chromosome 12, and it affects a protein that when it's defective, it builds up in nerve cells and causes serious neurological problems. So that is one where prevention, there's no known treatment or way to make it better. So if you have it, it's lethal most of the time because it affects the central nervous system. So autosomal dominant disorders, if you have just one bad allele, it causes the disorder. So only one parent needs to carry the allele. So if you have it, even if you're heterozygous for the trait, you are affected. So if you had an unaffected mother and an affected father, each kid has a 50-50 chance of being affected or unaffected. So there's no carriers because if you have it, you're affected. So unaffected persons don't transmit the disorder. Some of these conditions become evident later in life. That's that delayed lethal genotype. So the allele for the disorder may have already been passed on to the next generation before diagnosis of the disease in the parent. So that's Marfan syndrome, polycystic kidney disease, Huntington's. So if you have the affected gene for Huntington's, you are going to have Huntington's disorder. And if you don't have the gene, you'll be unaffected. So autosomal dominant disorders are a little more straightforward because if you have the gene, you're affected. There's no carrier. So those are the ones I just listed. Ehlers-Danlos syndrome is not listed in the book, but it's one that we're seeing more and more in therapy because of the effects on the body. It's a group of disorders that affect connective tissues supporting the skin, bones, blood vessels, and many other organs or tissues. So there are defects in the proteins that make up the connective tissues that cause the condition, basically. So there's several different subtypes of EDS, and they can range from mildly, the symptoms can range from mildly loose joints to life-threatening complications like aortic dissection if it's affecting the connective tissue around the arteries. Interestingly enough, EDS can either be an autosomal recessive or an autosomal dominant. They're different variants of the genetic disorder. So there's I put a page in the module. There's a link to some information on the internet about Ehlers-Danlos. We are seeing a lot more of these. Well, probably we've seen these people all along in PT because they had hypermobile joints and subsequent orthopedic issues because of it. A lot of times they get early osteoarthritis because of wear and tear on their joints. But now we're seeing these people diagnosed more. And so the way we treat them in PT is we work on stabilization. So we want to strengthen their muscles to stabilize the joints because the connective tissue isn't doing its job. A lot of people with EDS who don't know it end up having a lot of orthopedic surgeries and things, and it's unfortunate because they don't heal as well either. So X-linked disorders, a recessive X-linked, the allele is carried on the X chromosome but not the Y. So it's manifested in heterozygous males because you don't have the matching unaffected gene on the Y chromosome. You would need another X to have that happen. Heterozygous females are carriers. Homozygous recessive females may be affected. So if you had the right combination, you could end up with affected females. Inheritance of X-linked disorders may appear to skip generations because of that carrier-daughter effect. And the examples are Duchenne muscular dystrophy and classic or type A hemophilia. So the probabilities for an X-linked disorder are a little bit different. You're either, for sons, it's 50-50. You're either going to be affected and you don't pass on the gene, or you're going to be, and you have the disorder, or unaffected and you don't pass on the gene. For daughters, none of them are affected, but 50% of them, or there's a 50% chance of being unaffected and you don't pass on the gene, and there's a 50% chance of being a carrier where you pass on the gene. So some of the X-linked recessives, like Duchenne muscular dystrophy, you are rarely going to have an affected father and a carrier mother. ending up with an affected daughter because the disorder is such that the person doesn't live long enough to reproduce. So it's, I suppose it might, it just doesn't happen because of that. So 50% of sons affected, or each son has a 50% chance of being affected. And none of the daughters are going to be affected, but each daughter has a 50% chance of being a carrier. So with an X-linked dominant, heterozygous males and females are both affected. There's reduced penetrance in females, meaning the trait is not expressed as much because they have that good copy of the gene on the other X chromosome. You're just not getting it expressed as much. Fragile X syndrome is an example of an X-linked dominant disorder. It's the most common genetic cause of cognitive defects. So the effects of X-linked dominance are variable and related to the extent of the mutation of the allele. So it just lays out a little bit differently because it's dominant, if you have the gene, you're going to be affected to some extent. So single gene disorders include the autosomal recessive disorders, cystic fibrosis, PKU, sickle cell anemia, Tay-Sachs disease. X-linked dominant disorders, Fragile X is the big example of that, causes cognitive problems. X-linked recessives, color blindness, which isn't really a disorder, it's just an effect. Duchenne muscular dystrophy, and classic or type A hemophilia. I just thought this graphic was interesting for someone with color vision deficiency. They see all that. So I guess the moral of this picture is if you're going to give someone flowers who has color vision deficiency, give them yellow flowers because they won't appreciate the other ones, or they'll all look yellow. So genetic testing may be available prior to conception or during the first trimester of pregnancy, or they might test a newborn. Usually testing is recommended for people who have a family history of a specific disease, or if they had previous birth to a child with an abnormality. There are certain ethnic groups that have high risk for specific disease, and they usually want to test pregnant women who are over 35 years of age because there's more of a risk of errors during meiosis. So getting tested, it allows you to be prepared for what you are going to need to do if you have a child born with a specific disorder. So it's a good idea to be tested, especially if you know that you have a family history for the specific disease. So blood tests for pregnant women, there's one that's called the alpha-fetoprotein testing, and it detects several different types of potential disorders. The in utero testing, amniocentesis, and chorionic villi assay, it's a way of testing. You basically get a little bit of the amniotic fluid, and you can analyze it and look at the karyotype and see what's going on with the fetus. Neonatal testing, you can, right after birth, they test for excreted metabolites, so that's how they test for fetal ketonuria, and blood testing, and sometimes the sweat testing for people with cystic fibrosis or suspected cystic fibrosis. So genetic engineering is basically isolating, copying, and transplanting genes in microorganisms, plants, animals, and humans. It's used actually widely in agriculture to produce transgenic or genetically modified foods. Really, the ultimate goal is you have someone that's got a genetic disorder, you insert a normally functioning allele to prevent the disease. It's technically possible in humans, but clinical trials have not been uniformly successful, so this is something that is out in the future. It's not currently going on in human beings, but it's an idea. It's potential. Gene therapy identifies genes and proteins responsible for a condition. So a lot of times when you look up, like for example, you look up Tay-Sachs disease, and it says very specifically what the gene on chromosome 12, it It's called the hexagene, and the protein that it codes for, it's been figured out. And the idea is to determine how gene expression is controlled. The ultimate goal is to produce a gene that will, a drug that will inhibit or enhance gene expression, depending on the effect that you want to have. There's a lot of research that's been focused on cancer growth promoters that has resulted in these type of drugs. So the graphic has the adenovirus that's injecting the DNA into the cell. That's a possible delivery scheme for for gene therapy, because that's what viruses do anyway. They inject their DNA into the cell, and then they promote the replication of their own DNA. So if you could use that to our advantage for gene therapy, again, this is something that's not currently being done, but there's a lot of research going on and trying to do targeted therapy for cancer. So genetic screening and DNA testing, you could screen at-risk populations for a specific allele currently with the technology that we have. The biggest problem, though, is it costs a lot, and there are concerns regarding privacy and access to information. So they use DNA testing currently to identify paternity or forensic purposes, you know, who was at the scene of the crime. But we've all seen the sci-fi movies where it started out okay, but then people were being discriminated based on their genetic profile. So it's not just sci-fi, it could happen in real life. So it's something that we need to be cautious about. Even though it's available and it could be done, we have to decide if there's a really good reason for doing it. So designer drugs or proteomics, it's really trying to research the gene expression and characterizing proteins that are significant in a metabolic pathway for the expression of a particular allele. Some designer drugs act differently in genetically different individuals. So the idea is that you develop drugs tailored to an individual's genotype to reduce unwanted side effects of medications. This is being done in cancer treatment more than any other area at this point.